Hello students, welcome to my channel. Today we will discuss the topic central dogma. So what is the meaning of this topic we will understand in this video. But before the discussion of this topic you have to just understand what we will learn in this video. So DNA, that is the genetic material in most of the living organisms, and it help in the transmission of the characters from parents to offsprings, because DNA packed to form chromosome, and chromosomes have the genes, and these genes are transferred from one generation to another. Then how the RNA is formed from DNA and how the proteins are formed from the DNA, that we will discuss in this topic. So that is the central dogma. So let us understand. So central dogma. Firstly, the double helical structure of DNA. DNA have the double helical structure. This is the one strand and that is the another strand. There are the two strands of the DNA, and this double helical structure of DNA was given by that Francis Crick, Watson and Crick, the two scientists in 1953. And there are the some silent features of this model also, that both the strands are complementary to each other and they are made up of the sugar and phosphate backbone, and the bases are projecting inwards. What is the pH of the DNA helix? So there are the some silent features of this double helical structure of DNA also. So after that, the Francis Crick, he also proposed one model, and that is the central dogma. So what is central dogma? Means DNA जो है, DNA की रेप्लिकेशन से DNA बनेगा और DNA से RNA बनेगा और RNA से क्या बनेगा? प्रोटीन. तो ये एक जो साइकिल है वो चलता रहेगा. सो दैट इज़ द सेंट्रल डोगमा. सपोज दिस DNA, व्हेन DNA मेक इट्स कॉपी, अगर DNA से DNA बन रहे हैं, इफ़ देयर इज़ द फॉर्मेशन ऑफ DNA फ्रॉम DNA, then that process is known as replication. रेप्लिकेशन मीन्स कि उसकी कॉपी बन रही है, DNA से DNA बन रहा है, तो उसको हम बोलेंगे DNA रेप्लिकेशन. सो दैट इज़ replication. And this topic we will discuss in this chapter in detail also, how DNA replication takes place, what are the different enzymes for DNA replication, that we will discuss. After that, there is the formation of RNA from DNA. Can you tell me what is the difference between RNA and DNA? Only there are the two differences between the RNA and DNA. First, in most of the living organism, RNA, that is the single stranded, but DNA, that is double stranded. And in case at the place of thymine base pair is there, so at the place of thymine uracil is present. अगर आपके पास कोई एक स्ट्रैंड है, चाहे वो DNA का स्ट्रैंड हो या RNA का, तो अगर उस पर बेस पेयर्स लगे हुए हैं और कहीं पे आपको दिखाई दे जाता है यूरेसिल, तो आप वहीं से पहचान जाओगे कि दैट इज़ द स्ट्रैंड ऑफ RNA. दैट इज़ द मेन डिफरेंस बिटवीन DNA and RNA. And the second, in case of DNA, the sugar, that is deoxyribose, and in case of RNA, the sugar is ribose. RNA, that is more reactive as compared to DNA, and there are the different forms of RNA also, different functions of RNA also. RNA play the catalytic role, RNA help in the formation of the protein, different types of RNAs are also there. So there is the difference between the DNA and RNA. So when RNA is formed from DNA, and especially which type of RNA? mRNA, या आप सिंपल लिख सकते हो RNA. RNA is formed from DNA, तो उस प्रोसेस को हम बोलेंगे ट्रांसक्रिप्शन। और फिर इस RNA से, इस मैसेंजर RNA से क्या बनेगा? देन देयर इज द फॉर्मेशन ऑफ प्रोटीन्स। दैट प्रोसेस इज नोन एज ट्रांसलेशन। सो दैट सीक्वेंस इज नोन एज सेंट्रल डोगमा। अगर कोई भी सेल है, in the center nucleus is present, and in the nucleus the genetic material DNA is there, and from the DNA, if DNA is double, then the replication takes place, and from the DNA if RNA is formed, that is the RNA, that process is transcription, and from this RNA then proteins are formed. A polypeptide chain of proteins are formed, that is the proteins. So that process is translation. So this is known as central dogma, formation of DNA, from DNA replication, so DNA to RNA and from RNA to protein. This is known as central dogma. कैन यू टेल मी कि जो ये सेंट्रल डोगमा है ये कई बार रिवर्स भी हो जाता है। रिवर्स कैसे? कि जो ये DNA है, DNA से RNA बनेगा और RNA से क्या बनेगा? प्रोटीन। बट सम टाइम दिस इंफॉर्मेशन इज़ ऑल्सो रिवर्स, और उस प्रोसेस को हम बोल देते हैं रिवर्स ट्रांसक्रिप्शन। रिवर्स ट्रांसक्रिप्शन। सो लेट अस अंडरस्टैंड दिस रिवर्स ट्रांसक्रिप्शन. Now the reverse central dogma, or also known as Taminism. So why it is known as Taminism? Because it was discovered by the scientist H. Tamin and D. Baltimore in 1970. So what they observed? They observed in some viruses, because in the viruses RNA is the genetic material. So what they observed, in some viruses one enzyme is present, and that enzyme is reverse transcriptase. So what is transcription? Transcription means the process of formation of RNA from DNA. और अगर मैं बोलती हूँ रिवर्स ट्रांसक्रिप्शन तो क्या हो जाएगा? RNA से क्या बनेगा? DNA. And now I am saying reverse transcriptase. So this transcriptase, that is the enzyme. So in these viruses the reverse transcriptase enzyme is present, and what this enzyme do? This enzyme can form DNA on RNA template. ये एंजाइम DNA बना देगा RNA से, और इसीलिए हम इसको बोल रहे हैं रिवर्स ट्रांसक्रिप्टेज। बिकॉज जो ट्रांसक्रिप्शन होती है वो तो DNA से RNA बनता है, बट रिवर्स हो जाएगा तो RNA से क्या बनेगा? DNA. सो फॉर्मेशन ऑफ DNA from DNA, that is the replication, DNA to RNA transcription, and RNA to protein, that is translation. That is the central dogma. But the reverse of it is that if RNA is formed, if DNA is formed from RNA with the help of the enzyme reverse transcriptase, is known as the reverse central dogma, or this process is known as reverse transcription. So this is the most common in case of the viruses, and these viruses can make DNA by the RNA template, and it is the reverse flow of information, because the linear flow of, or the single flow of information, that is the central dogma. But if it is in the reverse direction, then we can say reverse flow of information in case of some viruses. So these two scientists proposed this reverse transcription so they got the Nobel Prize for this activity also. And this is very essential to study the cancer, because in case of cancer, what happened, there is the abnormal cell division takes place, and finally that abnormal cell division give rise to tumor. So this we can means this phenomena can also help to study the cancer. So that is known as the reverse transcription. And in your NCERT, one question is given, if this central dogma is reverse, means it, if it is in the opposite, so that is in the opposite in reverse transcription. So that is about central dogma and reverse transcription. Thank you.